Now that you've seen how the language of circuitry is written and used to power electronic devices such as your computer, the last step is to see how the language of a cell can be written, constructed, and processed using the same system of logic gates. Similar to the last video, where we started our journey by explaining the individual parts of the transistor and how their connections lead to information processing in a computer, we'll begin this video by looking at the key elements in the flow of information in a cell. Thankfully, you'll see that we've described a number of these elements already over the past few weeks of the module. Throughout each lecture, I've emphasized the analogy that we can think of a cell just like a self-replicating machine, where the information blueprint in the DNA is accessed and used in ways that lead to the manufacture of the pieces needed to build the cell in composite parts. The parts can then be assembled into larger functional complexes, and finally they are distributed throughout the cell to perform specific tasks. The culmination of this flow of information throughout the world of a cell is ultimately what permits cellular life to exist. Now that the science has come to a stage where we know all of the sequence information in the DNA blueprint for thousands of different organisms, and we've made great strides in knowing precisely how to write, construct, and decipher the information blueprint using the central dogma, this means it should be fairly straightforward, in theory, for how we could go about using the basic rules and logic of circuits to construct living cells such that they process the information in a specific way that we want. Although the field of genetics has come a long way, there are still a number of things we don't fully understand, and the precise details will be constantly revised over the coming years. Despite this gap in the specific details, we can still use the fundamentals of circuitry to envision how the information processing in a cell could be constructed and controlled using the basics of circuitry. We've already discussed a number of the key elements that are essential for information processing in a cell before, even down to the level of things such as the on-off switch and input information. For example, recall how every gene is a linear sequence of DNA nucleotides with a beginning and an end, and the part that controls whether the gene is actually expressed or not is called the promoter. If we were to apply the language used in circuitry, we could think of the promoter region as the on-off switch. But what then is the input for the switch? What is the input for a gene? Hopefully you can see for yourself how an example of an input signal could be any of the essential factors that are needed for gene transcription, such as the RNA polymerase. Given these examples for the switch and the input, you should be able to envision that the options for an output signal in this type of circuit could be either the mRNA or the protein itself. Recall from previous lectures that what makes each cell unique in your body comes down to what genes are actually being used by each cell. Using this basic diagram for a gene, we can see where the switch is located and begin to ask how it might be turned on and off. To begin, I could ask the following. Since we have over 20,000 genes in the human body, does that mean we have 20,000 different types of switches? I could also turn this question around to ask instead, do we have 20,000 RNA polymerases that would require 20,000 different switches? The answer to both of these questions is no, and the reason why comes back to the basics of evolution. Because of random DNA mutation and its selection over time, we can see that the cellular machinery has been adapted to eventually take new roles, rather than having a cell invent something entirely new from scratch to do a certain function. This is also true of the gene-copying enzymes, the RNA polymerases. Their universal function is to transcribe genes, so at their basic level, they have evolved over time to recognize certain elements in the promoter regions of DNA. This means in the simplest case that we could look at whether the RNA polymerase is present or not as being the first type of input signal that would be able to turn the switch on for a gene. If we look at a number of different genes and the precise sequence of DNA nucleotides in their promoters, we can see the ideas presented in the last slide visualized here, where there appears to be a related set of sequences. The areas where this occurs have been marked using pink and yellow highlighting. The take-home message is for you to appreciate that the switches seem to have a similar look to them, which allows the RNA polymerases to find them easily. To get a clearer picture of how to summarize gene expression using circuitry, first recall the basics of gene transcription shown here. Once again, let's take the simplest case, where the presence of the RNA polymerase alone is enough for gene expression, and thus it serves as the input signal. In this example, we would say that the truth value for the polymerase is 1, because it is present and it has a place to bind to. The transcription of the mRNA and/or its subsequent translation to a protein could then yield the final output signal for the circuit. Hopefully, you can immediately see that access to the switch could be a major factor in controlling whether genes will be expressed or not, based on the simple fact that the RNA polymerase needs to be able to bind to the DNA in the promoter to begin with. Some of the factors that might regulate the switch and ultimate transcription of a gene are explained here. The key take-home message is to understand that gene transcription is highly complex and a number of different factors can control it. This diagram shows how it is conceivable to have many input factors that might all be needed for the switch to be pushed into the full on position. Although the number and combination of inputs may vary, notice how we consistently envision that the only way for the output signal to be generated is for the RNA polymerase to be present and active. Building upon this general idea for how the circuit in a cell is constructed, we can see an example for how we could envision the AND gate logic here. Let's just assume at the beginning that the gene is off and all of the input and output truth values are 0. To start constructing the AND gate, hopefully you remember that it can have two possible inputs, and both are needed for the circuit to switch on. In our diagram, we can envision that there are places in the DNA where these two inputs will bind and control the switch. Next, think of this gene Y as being something that is needed only at a particular time. For example, let's say the gene Y encodes a protein that helps to break down the sugar lactose, so it can be used as a source of energy. When the cell is exposed to lactose, it functions like a signal that can be sensed by certain parts of the cell, such as this activator protein shown here by the green glow in the cytoplasm. Upon binding to its signal, the activator protein will move into the nucleus of the cell, where it will bind to the DNA of specific genes. We can see the movement of the activator protein into the nucleus quite clearly in this video. When the activator protein binds to its signal and subsequently to the promoter of gene Y, we assign an input truth value for this factor as 1. Please notice that there are two-way arrows to show that the activator protein can also release the signal, meaning there isn't a permanent change to the protein. Knowing that gene transcription always requires an RNA polymerase to do the copying to begin with, this would serve as the other input value for the AND gate. For comparison, we can also show the basic logic symbol for the AND gate that would describe this particular example. Although both inputs are clearly needed for the AND gate to work, hopefully you can see that any RNA polymerase that is present could bind to the polymerase binding site, but it takes a specific signal and a specific activator protein to turn on the switch for this particular gene Y. This is why you see the overall logic in this example has been summarized with the emphasis on this factor only. Just as there are proteins to activate the expression of genes, there are also proteins designed to repress or block other genes from being expressed. In the second example, notice once again that we have a place in the DNA where a factor can bind to the switch, and when the protein receives its signal, this causes it to bind to its element in the DNA. Unlike our last example, we can see that this protein functions as a repressor, meaning there is no transcription or output from the circuit. Likewise, you can see the overall logic summarized for this example with the emphasis on the repressor only, because in reality, it doesn't matter whether the RNA polymerase is present or not, right? If the repressor has received its signal and it is there to bind to its element in the DNA, the circuit is categorically considered off. If we wanted to use the basic logic gates to describe this flow of information, couldn't we use a NOT gate? However, notice that the binding of the signal to the repressor is a reversible process, similar to what we saw in the last slide. When we begin talking about one thing binding to another, whether it be this initial signal or whether the protein itself is binding to the DNA, hopefully you can begin to see that this activity can be measured. We can measure the amount of time the repressor normally binds to its signal, and we can measure the amount of time it is bound to the DNA. This concept is what we refer to more generally as the kinetics of binding, similar to how this word might be used to describe the rate of reactions in chemistry. Let's now assume that the binding of the repressor to the DNA or the signal has exceeded its time and the repressor leaves the DNA. When this happens, we could assume, unless we're told otherwise, that the RNA polymerase is available to bind to its element in the switch, and the gene is subsequently transcribed and translated. This is how we could keep using the original NOT gate symbol and just alter the input truth value. The repressor example in the previous slide was described using the simplest gate logic, but this required making an assumption about the RNA polymerase, and it didn't capture all of the information that could have contributed to the overall logic in the circuitry. Can you think of other ways to write the gate logic for the repressor example that might allow you to capture more information? For example, one truth table that could be constructed is shown here, where the repressor bound to its signal and the RNA polymerase are the two input variables. Unfortunately, this truth table doesn't look like any we've seen previously, right? This should indicate to you that the gate logic will not be constructed using a single gate, but that it might be formed from a connection of multiple gates. We could go even so far as to try and describe all of the information in a single truth table, but you should immediately see that as more variables are added, the number of input combinations increases exponentially, where we have 4 possible outputs with 2 inputs, and up to 16 outputs with 4 inputs. Fortunately, we can immediately see that only certain combinations will ever lead to an output truth value of 1. For example, we must have the RNA polymerase present with an input truth value of 1 for the output truth value to ever be 1. We'll expand upon this idea in the next lecture series when we discuss stability of a system and how to construct larger networks called Boolean networks. Hopefully you see from that last example that cells can be constructed with simple on-off switches, just like an electrical device. All we need is something that keeps the circuit off, like a repressor, and the output could be anything. In this next example, we're going to see that the output is a fluorescent protein called cyan fluorescent protein, or CFP for short. The CFP gene that encodes this protein has been constructed into the bacteria that you will be using in the lab practical over the next few weeks. Thus, it is helpful for you to see how their circuitry is constructed. We can see from the top picture that in the normal default condition, it differs from our last example slightly, where this repressor is bound to the DNA regardless of whether it has received a signal or not. This doesn't mean that our repressor doesn't have the capacity to be responsive to things though, right? We could add a particular chemical that is known to bind to it, such as IPTG, and this would lead to the repressor being inactivated. Remember that proteins can be flexible and that binding to substances can physically change their shape in ways that prevents them from subsequently binding to other things, like DNA. We could envision this as how the repressor is inactivated by IPTG in this example. As a result, the polymerase protein is able to read the gene, and we ultimately see an increase in the fluorescence output. Another difference you might see with the previous example is that the binding of IPTG is not reversible to our repressor. This means that over time, as every repressor molecule binds to the IPTG we've added, we'll see an increase in the fluorescent output until it reaches a maximal value. If we had a way to measure the output fluorescence, we could quantitatively analyze how the CFP gene expression changes over time, right? This graph shows precisely this idea and what type of data you might find when the principles of circuitry are applied in the research laboratory. This is sort of an idealized output, but it's actually from an experiment published over a decade ago, where a simple circuit like the one shown here was engineered into a cell, and then this is the simple output. Over the next few weeks, you will be learning how to apply and use these very same ideas in your lab practical. The final piece of information you should appreciate at this time about simplifying a biological organism into a system of circuits is this. Each cell has its own set of information and cues that it is responding to in space and time. If we look at a population of bacteria and we engineered them all with the on-off switch seen here, we cannot expect that all of the bacteria will activate expression of the gene at the same rate and subsequently express the protein at exactly the same time to become fluorescent at exactly the same moment. If they did, we could model the fluorescence as a step function. By looking at this graph, we know this simply isn't the case. In biology, where there is a digital output, we find that it is not exactly digital in reality. Despite this drawback, hopefully you can appreciate that thinking of it in this way can still be useful when we want to begin constructing and modeling living organisms. In the last video of this lecture series, we'll see a few real-world examples for how a cell has been engineered using the principles of circuitry.